Ready to go.
Like compare it with a lot of
Hello everyone, it's my pleasure to introduce you to our guest speaker, Dr. Haiyan Xu today. Dr. Xu is an associate professor of medicine at Alpert Medical School of Brown University, and she also serves at the Hallett Center for Diabetes and Endocrinology in Rhode Island Hospital. Dr. Xu studied basic medicine, more specifically biochemistry and molecular genetics at Beijing Medical University in China, from where she received her Doctor of Medicine and Master of Science degrees. From 1994 to 2000, Dr. Xu pursued a PhD degree majoring in medical science at Howard. Then she continued her postdoc research for 1 year and being a research scientist from then on in the Department of Metabolic Disease Biology at the Millennium Pharmaceutical till 2004. Then she conducted researches on metabolic disease at Alpert Laboratories till 2005. Dr. Xu joined the Warren Alpert Medical School of Brown University in 2005. She's now the associate professor in the Hallett Center of Diabetes and Endocrinology. She's involved in the research of the molecular mechanism in obesity-related insulin resistance and type 2 diabetes. This includes Adipose inflammation that disrupting insulin signal underlying obesity, novel factors that regulating adipose energy metabolism and the mechanisms of dysfunctional hemostasis of liver glucose. She made big discovery of obesity, including adipose macrophage infiltration, which was honored as one of the milestone discoveries of the Journal of Clinical Investigation. She was awarded the Medical Sigma Harvard Radcliffe Chapter Scientific Recognition Award, Edgar Hoover Award in Biological Science from Harvard School of Public Health, and Bruce Celia Award for Research Excellence by Rhode Island Hospital. From 2005 to 2013, the program in Brown University is very productive and have published more than 28 peer-reviewed papers. So everyone, let's give a warm welcome to Dr. Haiyan.
Thank you very much for the introduction. I'm very glad to be here today. I want first thank Dr. Wu to give me the opportunity to present work for the graduate students here. And so as introduced by a fellow student, my work has really been focusing on obesity-related metabolic disorders. So my work, focus on like adipose and liver, but today I'm only talking about the role of adipose inflammation, what it plays in obesity-related insulin resistance. So I think probably everybody is really familiar with this kind of slide. You know, the obesity trend during the past quarter century, that how the incidence of obesity is going up so rapidly. Like back in 1990, if you look around the country, no state has obesity incidence more than 15%. But today, no state has the incidence of obesity of less than 20%. Well, here's Texas, 30 to 35, not the worst, but yeah, doing okay. And as you all know, that obesity, is a risk factor for the development of many chronic diseases, like typically type 2 diabetes or dyslipidemia, hepatosteatosis, cardiovascular disease, hepatocellular cancer. You know, it's not by no way the complete list, but particularly obesity is so highly correlated with type 2 diabetes. Have you heard the word diabetes to describe the relation? And type 2 diabetes is the form of diabetes, insulin resistance that characterizes type of diabetes, in contrast to type 1, which is insulin deficiency. So insulin is the most important hormone to regulate blood glucose metabolism, and its major target tissues are adipose, muscle, and liver. And the resistance in peripheral tissues plus abnormal insulin secretion of allies are the major reasons leading to the development of type 2 diabetes. And the prevalence rate of diabetes is really high now. It's up to like 9.3%, not 93, 9.3% of the population. So it's a huge public health problem. So yeah, I'm trying to separate the hypothesis of the mechanism of obesity-related insulin resistance in this slide. Basically, the development of obesity is a simple equation: energy intake exceeds energy expenditure. And the excessive amount of energy is normally deposited as triglycerides in adipose tissue, white adipose tissue. The storage capacity of adipose tissue is exceeded, and the energy can be spilled into circulation or other tissue in the form of like free fatty acids. And free fatty acids being well characterized to induce resistance to insulin in other tissue like muscle or liver. So it's a consequence of which there's less oxidation or less glucose utilization by muscle. Liver usually is the organ that pumps out glucose, in times of energy shortage. But in this case, because insulin resistance, there's more glucose to be produced by the liver. So as the consequence, because insulin resistance, and beta cells have to work harder to produce more insulin to compensate. And then hyperinsulinemia will occur that indicates the existence of insulin resistance. But eventually, beta cell cannot follow up with the need of the body and fails to produce enough insulin to control the blood glucose level into the normal range, and eventually the blood glucose level is out of control. That's how type 2 diabetes occurs. So why am I so interested in adipose tissue? Because one thing about obesity is all about the pathological expansion of adipose tissue. So this is the only tissue in adults that can seem to have unlimited capability of expansion. So the expansion of adipose tissue comes in two ways. So in humans, it's mainly enlargement of existing fat cells in volume, and a small portion is about new adipogenesis, production of new adipocytes. But in rodents, I think both forms occur. And adipose tissue is not only an organ for energy storage, it also actively secretes hormones like leptin or adiponectin to regulate energy homeostasis. So it also secretes inflammatory cytokines or chemokines, and also, it regulates whole body energy homeostasis by storing energy when there's surplus, but release energy in the form of free fatty acids when energy is in shortage. So, like, I want to briefly talk about like the types of adipocytes. So, what I just mentioned is the white adipose tissue. There are two types of adipose tissue in humans or rodents. White adipose tissue, this is histology of how it looks like of white adipose tissue, and these cells like big single lipid droplet in the nucleus of the side of the cell, and in contrast, there's also brown adipose tissue. It's a tissue that dissipates energy as heat. So it's rich in mitochondria, and there is a special protein called uncoupling protein 1 that's highly expressed in this cell type, as you can see in color. And so brown adipose tissue is desired, especially like in 2009, there were 3 publications in New England Journal of Medicine indicating there is brown adipose tissue in humans, because in the past it was thought, okay, brown adipose wasn't relevant in human physiology. So, then stimulating brown adipose function could be one approach to prevent obesity. So lately, there's also another type of adipocyte called beige adipocyte that's in between white and brown. It can be induced, has same cursor as white adipocyte, but not brown. And you can see, it expresses UCP1 and urea is interspersed among white adipocytes. So my research career, actually really started at a pharmaceutical company. So after I finished my PhD training, so I wanted to really look for solution for treatments of obesity-related metabolic syndrome. So I decided to join Millennium Pharmaceuticals. At the time, they were doing really active research to look for novel targets. So my job was to find novel targets that's related to obesity-related insulin resistance. So we use several approaches like transcription profiling, or dismantling, by looking at tissue distribution patterns, or functional screening. So, I came up with, some, interesting results with each of the approach. So, like, discovery of adipose, macrophage infiltration from group number 1, and by looking at, human kinase distribution pattern of more than 300 kinases, I came up with 2 that's abundantly expressed in adipose tissue. And by using a functional assay, looking for novel genes that can repress the function of insulin by using, type CK promoter as approach, I came up like 2 MAP kinase phosphatases as mediators of insulin resistance. So, today I'd like to touch the topic of adipose inflammation, and later I'll talk about most recent and published work about novel kinases we're working on. So how we discovered macrophage filtration. So, at the beginning, I had no idea what's going on. So we decided, okay, why don't we look at gene regulation patterns in, all of insulin target tissues, and plus something else. So we did like profile A tissue by transcript profiling. And it was amazing that we found that adipose tissue was tissue with most dramatically upregulated genes that mostly are macrophage-related and inflammation. And that occurs in like both diabetes obese mice and genetically obese mice. And that led us to hypothesize that, because presence of macrophage usually indicates chronic inflammation. So we were thinking, okay, maybe macrophage-mediated chronic inflammation in adipose tissue could be one mechanism why obese people tend to develop insulin resistance. So that's how we found that there are macrophages in obese adipose. So this is a slide of lean mice adipose and obese adipose. You can see there's a lot of standard basic lipo80 is a markable macrophages. And people now give a name called crown-like structure because these macrophages usually surrounding dying adipocytes. And when you isolate, the stroma fraction, which is a non-adipocyte cells that you can isolate from adipose tissue, you can see that there's a lot of giant macrophages with multi-nucleotides in the adipose tissue. So later on, it turned out that this finding contributed significantly to the field of metabolic inflammation. So following that, there was a lot of important work that showed up in top tier journals like Nature Medicine, and people found there are not only macrophages, there are a lot of other type of new cells that can accumulate or decrease in adipose tissue in obese rodents, at least, like neutrophils, mast cell, B cell type of T cells. And then also people found that, what happens is that there was classically active macrophage accumulation in obese, adipose, but which is more pro-inflammatory. And alternative active macrophages, which are anti-inflammatory, they actually they diminish. And also other good cells like regular T cells, eosinophil, natural killer T cells, they all decrease. So, I'm really just focusing on macrophage because right now I don't believe which type of cell migrate into adipose first, or which the exact order is not determined. But because a large quantity of macrophage in fat, and the fact that there's such a high amount of inflammatory cytokines, they have been put at center stage. I believe there are more papers about macrophage than other immune cell types. So, Dr. Anthony Brantley lab at Columbia University, they did same finding simultaneously with us, so we published all together in the same issue of JCI. But they also show that this crown-like structure also exists in human fat. And macrophage marketing expression is positively correlated to BMI and adipocyte area. And Dr. Grunberg's lab at Tufts University, also found that the macrophages, labeled as M, tend to, surround dead adipocytes. Then a lot of publication came up late to prove that if you manipulate macrophage content in fat tissue, like, by different genetic models, if the adipose macrophage content is decreased, and usually the obese mice tend to be more insulin sensitive. And if you conditionally depletion macrophages, and it would rapidly normalize insulin sensitivity in obese mice. And if you overexpress, factors that can attract more macrophages into fat, and mice tend to be more insulin resistant. So the large amount of literature data support the concept that adipose macrophage accumulation can cause insulin resistance. Also, there's some human studies that by diet restriction or bariatric surgery, and then when you lose, your fat mass, there's also usually associated with an improved inflammatory profile and decreased expression of macrophage genes. And the only class of insulin sensitizer that has been used to type 2 diabetes on the market, it also has potent anti-inflammation effect by decreasing expression of macrophage genes and macrophage activity. So we later also did our lab, try to avoid macrophage by chemical approach. It's called collagen liposome that can specifically kill macrophages. And by looking at obese mice, these are different fat depots. You can see crown-like structures, and the treatment effectively reduced the crown-like structures. Well, in lean mice, this did not seem to have an effect because there are very few macrophages there. And it does not change body weight or blood glucose or insulin. But in obese mice, when there were large amount of macrophages, when that got depleted, we had decreased blood glucose levels, as well as plasma circulating level of glucose, indicating this approach did improve glucose homeostasis. And because we did, intraperitoneal injection of collagen liposomes, and what we found is that, these can also be, absorbed by liver. And it was very interesting. We found that hepatocytosis were significantly improved by seeing less, lipid accumulation in the liver. All right. So, why macrophage come into the fat tissue? There has been extensive work on this topic, and the people come up with all sort of theories, like hypoxia, rapid fat mass expansion, not enough vessel development, there's lack of oxygen, and then adipocyte death, and macrophage come in to clean it up. Well, activation of master inflammatory kinase, like IgA beta or JNK, where you increase secretion of monocyte chemotactic factors, or decrease secretion of anti-inflammation adipokines. So personally, I favor the initial macrophage infiltration is caused by increased secretion of monocyte chemotactic factors, because my hypothesis is that adipocyte is the first type of cells got stressed when there's overload of nutrients or lipids, and then somehow you send out a signal and asking for help. If macrophage coming in, my hypothesis must be increased secretion of chemotactic factors, because in immunology, this is a way that how immune cells are attracted to cellular inflammation. So we just profiled, the mouse homologue of all of known chemotactic factors. So we found that there were at least 6 monocyte chemotactic factors are significantly upregulated in adipose tissue or dye-induced obesity. Macrophage chemotactic protein 1, 2, 3, macrophage inflammatory fat protein 1 alpha, macrophage-related protein 1 and 2. So yeah, their expression level are significantly upregulated in high-fat diet fat mice. And this upregulation seems to occur in the adipocyte fraction. You can really take adipose tissue, separate the adipocytes from everything else. So it's occurring specifically in the adipocyte fraction, not the other fraction. We call stromal vascular fraction. And when we perform like in vitro chemotaxis assay, each one of them can attract macrophages. But when adding together, there was an additive effect. So I think that it is more than one factor that's attracting macrophage to come into fat tissue in obese situation. Then we also did a little mechanism study, say, how these chemokines are upregulated. So we look at several pathways, And what we found that IKK beta is the most important kinase that's inducing the chemokine expression. For example, when we tried to treat fat cells with free fatty acids, so all these chemokines are up, but when we inhibit IKK beta and kappa B pathway, all of them are significantly downregulated. And when we knock down IKK beta, we got similar results. And when we opened up wild type, we were able to increase the production of these chemokines, 5 of them, at very high level. And this is constitutionally active IKK beta that can promote production of chemokines at even a higher level for most of them. And when a kinase dead, IKK beta was expressed, there was no effect. When you look at insulin signaling, the constitutively IKK beta significantly reduced phosphorylation of KT, which is a very pivotal point of insulin signaling, the phosphorylation, on both threading 308 and serum 473. And well, there are a lot of reports in literature. This has been described as one of the center-mediated obesity-related insulin-resistant inflammation. And we found, yes, in the expression of this kinase, got increased pretty quickly, even 1 day after high-fat diet. And when you treat the adipocytes with free fatty acids, which significantly reduce the phosphorylation of AKT, but the adipocytes liking IKK beta, you can see that phosphorylation is much higher than the wild type without treatment, and still significantly improved compared to wild type under treatment that has a insulin with that. And then we said, okay, well, we will do an animal model, so we will overexpress IKK beta constitutively adipose tissue. Do we have a model of adipose inflammation and insulin resistance? So we did the transgenic mice. And yes, indeed, in the circulation, the mice had much higher, significantly higher, much higher. Significantly higher level of at least 5 monocyte chemotactic factors. And we look at the percentage of macrophages, pro-inflammatory macrophages, represented by CD11C, and they are significantly increased. But there is one issue with our model because inflammation usually consumes more energy. So here we are artificially creating an inflammatory model without the context of obesity. What happens is that the mice were actually leaner under both child diet or high-fat diet in both males and females. Adipose tissue actually was smaller in both genders, either on child or high-fat diet. And the histology will show you that, yes, under normal diet, since the transgenic mice have a little smaller adipocyte, high-fat diet, yes, there were some crown-like structures, but transgenic mice, yeah, they have a lot more of these crown-like structures indicating macrophage infiltration. So yeah, that seems true for both. Well, not all fat depot was subcutaneous fat depot, but as I mentioned, but it seems this activation of this pathway burned off more fat storage, as exemplified by smaller adipocytes and less accumulation in brown fat and liver. So as a result, surprisingly, actually we did not get systemic insulin resistance. So this is not surprising. I want to point out several models like Ron Kahn's lab, like Johnson. They did adipose insulin receptor knockout mice, which is no insulin signaling in adipose. You see highly insulin resistant, right? But then on the other side, there is no impairment in fat development. There's less fat, you know. And overall, the benefit of being lean override the harmful effects of local insulin resistance. So mice will live longer and insulin sensitive overall. That's what I saw. You know, we had a little lower blood glucose in those gender. And when you test mice by injecting glucose, it seems transgenic cleared up more quickly than the wild type control, where you know they respond more to insulin than the control, and the most high fat and you know chow feeding situation. So that made us to think that you know what's also having inflammation in obesity, why it happens. So one hypothesis is that it's one way. One is the body send out signal saying now I'm really fat. I need you know all the factors coming in to try to secrete inflammation inside of to induce lipolysis to make fat mass smaller. So it might be one approach to limit fat mass development. But we always see, you know, where inflammation occurs, you see insulin resistance locally in the tissue. But if this is successful in reducing fat mass, when systemic insulin resistance does not necessarily occur. So what I think, if I induce the expression of AKT beta activity later in obesity context, I would see insulin resistance systemically. Okay, so that's personal opinion. So adipose inflammation, yes, it does induce insulin resistance, definitely in the tissue. If it does not affect fat mass, you probably see systemic insulin resistance and hyperglycemia. But if it is reduced fat mass, like our model, you know, because of obesity lipids, you may not see systemic insulin resistance. And yeah, so I haven't talked about quite a bit about how you know the inflammation pathway, and now I'm switching a little bit gear about like is there anti-inflammatory pathway? You know whether that's decrease in obesity. So okay, compared to this aspect, this is less studied. Later, people have report some anti-inflammatory effect of AMPK-alpha-1 or SIRT1 in adipose, but by looking more closely, these are mostly occurring in macrophages in adipose tissue. So we are really interested in studying, the anti-inflammatory mechanism in adipocytes. So in the beginning of my talk, I mentioned that we try to different approaches to pull out novel genes that I came up with SNRK, sucrose non-fermenting related kinase, as a potential suppressor of adipocyte inflammation. So initially, we were attracted to it because it was so highly expressed in adipose tissue versus other tissue. Then later on, we thought, okay, we have some interesting gene regulation patterns, but we had no idea what it does. If you look at the literature, you essentially have no information about the function of this enzyme in mammals. So these are the tissue distribution patterns of SNRK in human and mice. So in human, it's highest expressed in white adipose tissue. In mice, it's highly expressed in both white and brown adipose tissue. So we did epi-western of the kinase, compared about 10 tissues. So the protein is mostly abundant expressed in white and brown adipose tissues. So it's ubiquitously expressed in many other tissues. And we found that this kinase expression is significantly reduced in both genetically obese mice and in diet-induced obese mice. at both gene level and the protein levels. And what's interesting is that we found when we separated adipocytes from non-adipocytes, this reduction seemed to be occurring significantly in the adipocytes, and not affected in stromal vascular fraction. And the expression level, this kinase is higher in adipocytes than the stromal fraction anyway. So first experiment, functional one we did was to knock it down in the adipocytes by interfering RNA first. So you can see the gene and protein are significantly reduced. And the phosphorylation of the two master kinases that can activate transcription of numerous inflammatory factors, the phosphorylation level was significantly increased. And we saw significantly increased release of glycerol, which outcome for lipolysis. So lipolysis is a process of, degrading triglyceride and release free fatty acids, secretion and glycerol. And we saw increased expression of pro-inflammatory cytokine TNF-alpha, so IL-6, and we saw increased production of macrophage, chemotactic proteins, and on the other hand, insulin sensitizing hormone like adiponectin is significantly reduced. Insulin responsive glucose transport 4 is reduced, and the genes involved in fatty acid oxidation, which indicating burning of fat, are reduced. So that made us to hypothesize that maybe this kind of important for repressing the inflammatory pathway. So then we derived the kinase assay, looked for the activity of this kinase. We found that in normal mice, the activity in rat adipose can be significantly decreased by fasting or high-fat diet, to the background level. And also in cultured adipocytes, yes, it's also significantly decreased. But if you stimulate adipocytes with this is acetylpyranol, is a beta receptor agonist that can really increase energy pressure. The kinase activity is dramatically upregulated. And so, so we decided to go ahead. Okay, it's interesting now, so let's make animal models. So first we tried with global SRK knockout, and they turned out to be parental. So they were born alive, but died within 24 hours. I'm not exactly sure why they died, but by looking at the stomach, they had no milk spout in the stomach, so maybe they were dying because they were not able to feed. So we had neomycin cassette inserted into the exon 3 and 4 plus a Lexi reporter. So you can see that the enzyme is actually expressed everywhere. At birth, the paths were about the same weight, but because the knockouts were not able to feed, the blood glucose levels were really low. So because there was no that fat yet at birth, so white active tissue development occurs post-birth. We were not able to study the function in adipose tissue. So so that we have to end up with characterizing the heterozygous first. So in the heterozygous mice, the ISRC gene were reduced by half in fat. We did observe increased expression of TNF-alpha, or the marker for pro-inflammatory macrophage CD11C. And at the same time, we saw decreased expression of transcription factors that's important for adipose development. So yeah, by histology, you can see that the fat cells are smaller, but they're more crown-like structure, and there is hyperglycemia upon fasting. Sort of, consistent with our hypothesis. And now we have adipose tissue-specific SNRK, not no mice, which is initial characterization. So the genes was, reduced about 70% in whole adipose tissue, but like I mentioned, the knockout is specific for adipocytes. So assume the efficiency of, reduction should be more potent when we isolate the adipocyte fraction, which we haven't done yet. Compared to control mice, which either only express Cre recombinants in fat or double upstream. So if you look at histology, yes, a consistent heterozygous mice. These mice have smaller adipocytes, but staying with what I480 is a macrophage specific marker. And it looks like, yeah, there's accumulation of macrophages there, but which not occur in control mice. And we tested for insulin and glucose tolerance. Yes, knockout mice seem to have glucose intolerance and are less sensitive to insulin injection. And yeah, we did try to profile the downstream signaling pathways. So because we have no idea how it signals, nobody has studied it. Nothing is known. So what we did was we tried to profile phosphoproteins that have been changed when the enzyme is either knocked down or expressed. So we came up with two proteins. One is EFND, is a subunit of elongation factor. Another one is HDAC, which is histone deacetylases, acetyltransferase. Okay. So yeah, when we did knock down the downstream potential downstream components of SNRK signaling, we were able to reproduce most of the phenotypes, like increased expression of pro-inflammation cytokines, particularly TNF-alpha, and chemokines, decrease of adiponectin, which is involved in fatty acid oxidation, or autophagy gene. And yes, knocking down HDACs, we had a similar effect, increase of pro-inflammatory cytokines, chemokine, decrease glucose, adiponectin, and genes involved in fatty acid oxidation, and increase of lipolysis or increased phosphorylation of JNK, although phosphorylation didn't increase as much as SRK knockdown. So that indicates, okay, we also try to use inhibitors of HDACs, and that give us similar effects as HDAC knockdown. In terms of cytokine expression, gene involved in glucose transporter, insulin sensitizing and connecting, or lipolysis. And when we look at insulin signaling, yes, the HDAC inhibitor did significantly reduce insulin signaling in terms of phosphorylating AKT, and both basal condition and also stimulated condition. But what triggers us is that this model seems different from our previous model. Although the adipocytes seem smaller, but the body weight of the mice actually increased about 10%. So we were thinking might be related to the fact that it's because the kinase can increase energy expenditure, deficiency of the kinase, although leads to inflammation in adipose tissue, it might also decrease overall body, energy expenditure. That's why the mice end up being a little higher So we look at a little bit at brown adipose tissue as well. So the kinase also significantly decreased in brown adipose tissue in obese mice. And it can be upregulated by cold exposure or lectin treatment, conditions that can increase energy thermostasis. So because brown adipose were already fully developed at time of birth, we were able to do some experiments that. Well, with episode SRK, we saw significant decreased expression of UCP1. So this is the most important feature of brown adipocyte, is uncoupled protein. And transcription factor that's important for that development. And Western blot and immunohistochemistry confirmed this finding. And by electron microscopy, we found that there's the density of mitochondria are decreased. So, and also there was very little lipid accumulation, and this is a larger amplification. Mitochondria are kind of sparsely distributed in the cytoplasm, and since there's some sort of abnormal structure in the mitochondria in the knockout mice. Yes, in the brown adipocyte, yeah, the SRK was completely absent. But when we're taking the brown adipocyte and do in vitro tissue culture, there is less adipogenesis. This is same with oil red oil to indicate lipid accumulation in fat cells. But for some state differentiated, seems they have larger amount of lipids and bigger droplets. So the overall content is the same. However, these cells lost the ability to respond to urbinate receptor agonists and induce adaptive thermogenesis. For example, when we treat the wild-type knockout brown adipocytes with this CL, a specific agonist receptor for agonist for base 3 adrenergic receptor, and the response of increased expression of UCP1 or PRDM16 or PGC1 alpha seems basically blunted. Western blot also shows there's definitely less UCP1 protein in these cells, hardly any response to the CL compound. And our collaborator at Novartis performed oxidation consumption experiment. And these SRK deficient cells seem to be, less efficient in oxygen consumption, so indicating less energy expenditure. So I think, so my last slide almost there. By looking at histology of brown adipose tissue, you can see that it's definitely different from the control mice. There's a less lipid accumulation. What I showed you before, that was day 1 pups brown adipose staining. So this is adult mice, about 6 months old. And there's a less staining of UCP1, but very interesting. I don't know what it means yet, but there's a heavy staining of FOH in this case. So maybe there's also inflammation going on in fat cells. And the gene was reduced about 80% in this case. So I think that's right now where we are. So we are continuing to figure out the phenotype of the adipose tissue specific knockout. And we're trying to understand the signaling mechanism in both white and brown adipose tissue, because it's totally unknown how this enzyme signals, what's downstream, what's the direct target, what's the substrates. And I think I'll stop here and thank all my collaborators and people who contribute to work at talk today. All right, questions?
We have plenty of time for questions, so let's open the floor up.
So I will start a question. Because follow up your the AKKO expression, because you showed that's a people side specific overexpression. So that basically just show that, and there's what I'm on to say is some other study, particularly they use the AKK, for example, like in the liver,
yes, I know that.
and also the brain specific knockout,
Yeah.
they basically demonstrate protective effects.
Mm.
So that could indicate some tissue-specific effects of inflammatory signaling. Can you just add some comment on
Yeah,
that?
I think what I heard is I know the same lab also did adipose specific IgG. They got similar results with ours, but they never published. So what I think is in this case, because it's a complicated, like the fat mass really is an important factor into what happens. So when they overpress in the liver, I don't recall that affected body weight or fat mass. But in this case, I also did another transgenic model before with overpressure TNF fat tissue. I got pretty much the same result, that smaller fat mass, and that is beneficial and for those harmful effects of adipose inflammation and insulin resistance. And also, I remember Dr. Ye at Pennington, he did NF-κ transgenic, which is downstream, substrate of IgG data, and his publication had very consistent results with ours. So by looking at all these models, I would think, yes, if inflammation occurs in the context of obesity, and it fails to shrink the fat mass, definitely you have insulin resistance, both local and systemic. But in the case if you do reduce fat mass, and not necessarily you have systemic insulin resistance. That's my opinion right now. So.
Thank you. Can I ask another question? Sure. This is just follow up of the adipose tissue infiltration, because that's your milestone discovery. It's very, very important. Since then, so there's a lot of studies to show the macrophage infiltration, particularly several years later after your discovery, they showed like a particular triple gamma knockout, particular specific knockout, and there's several others indicate. So what I really want to say is they basically mainly focus on macrophage polarizations in one versus
okay.
2. So any comments based on your research?
Well, based on my research, I actually haven't done a lot of alternate macrophage activity stuff, because after this initial publication, I left Millennium, went to Alpert, doing my liver enzymes for a couple of years. While Carol Lumen published all this alternative activation in JCI, the AG Chala, and did a lot of things, PDR gamma and alternate macrophage activation. So I saw that, okay, it was so highly competitive. There was no way when I come back, was in any position to compete with them. So I was like doing other stuff. But I did meet Anthony Fergie a few months ago, and he came to our place for the seminar. So it's funny because he said he spent a lot of time looking for this M1, M2 macrophages thing. If you know that he published a paper later 2013 claiming in cell metabolic, claiming that, okay, well, there isn't really M1, M2 thing. It's just purely the amount of quantity, the quantity of macrophage increase in the fat mass in this cell. Okay, that's basically contradictory to what A.J. Chala published before. So I don't know. I really haven't looked into it. Yeah.
Thank you.
Yeah.
So, very nice talk. I'm interested in your SMRK heterozygous mice.
Okay. Yeah.
Fine, but we should increase the inflammation.
Yeah.
I'm wondering if you have culture with strains for a longer time, because they have increased spontaneous tumors.
Oh, tumors. I don't know. I haven't done that. Like, I could, I guess. So, yeah, because
people they need APC background, see increased duration and
yeah, right. So the reason I didn't do a lot of work about heterozygous is I was criticized by some clients. Okay, now you have this enzyme expressed at half of the level in all of the tissues because it's not really specifically edible. So when I saw the question, okay, it may not be the consequence of what it does in that. So that forced me how to do edible specific stuff and thoughts on that. Yeah, I think that could be useful for developmental things because I have different phenotypes. For the heterozygous, they're thinner, but for the edible specific knockout, they're heavier. So that tell you that other tissues probably play a role as well. So.
The same question is if you knock out the SNRK mutants in your pre-adipocytes, you said that you saw less cells, but they have more fat. And you did a kind of cell cycle profile to see which cell cycle phases affect when you move SNRK.
Okay.
Similarly, to look at the continued knockout as in the liver also, you can see if the cell cycle affect.
Yeah, I think that's an interesting question. That could be done because my lab really hasn't done any cell cycle work. So, yeah, it's something we can think about. Yeah, but really, we have to touch that aspect. Yeah.
Other questions? I was curious about your comment on the fact that the macrophage infiltration seems to be centered around dead adipocytes. That would suggest that it's a necrotic type of death. How well could we possibly design interventions to regulate the process of cell death in adipocytes so that you're undergoing apoptosis as opposed to necrosis in order to reduce that macrophage infiltration?
That's a very good question. So I think people think that small necrotic deaths versus apoptosis. And people hypothesize that why they die because they burst. There are four reasons that they do that. So one way could be, you can genetic modificate, something like various transporters, people like prone to basic accumulation. Would that be better, if they don't die in the rest of the fridge, things like that. And I think, it could be that way, like do edible specific, like now everything that can promote the people side different information. So,
Okay. Anything else? If not, let's thank the speaker. Just a reminder, if you're in the class and you haven't signed the sheet already, please be sure to do that before you leave. And then we also remembered students, we have a lunch invitation with the speaker in room 126 right after this.